Good afternoon. My name is Helen Francis. I'm the Research Strategy Manager at the Department of Computer Science and Technology. I'd like to welcome you to the department's first Research Showcase event this afternoon, focused on healthcare. We've got a very interesting lineup of talks today. We're going to be recording the event so that should you have any colleagues who were unable to attend, they will be able to view the talks afterwards. Following an introduction from Professor Anne Copestake, our head of department, I will then introduce the first of our speakers for our lightning talks. This will be Sarah Morgan. Each of the first three talks will last for just 8 minutes, and following on from the talks, there will be an opportunity to ask questions through the Q&A functionality. Our final talk, AI Methods and Tools for Bench-to-Bedside Research in Cancer Medicine, is going to be given by 3 researchers. Each of these speakers will talk for 8 minutes, and because they're talking on a similar topic, we're going to have the question and answer session at the end of the talk. I'll now hand over to our head of department, Professor Anne Copestake, who is going to open the event. Thank you.
Okay, thank you very much, Helen. And I want to say that welcome to everybody who's joining us. This is a very new idea for us, and we're thrilled to have so many participants who have registered. And as I was looking at the screen, we had more and more people joining. So this is wonderful. This is going to be the first of a series of themed events. We're talking about different applications of computer science research. So all of these things involve exciting computer science research as well as exciting applications. We expect to see future events on areas such as education, climate and sustainability, and other topics. So there's a considerable history of healthcare-related research in the department. And what we're seeing today is an opportunity for the early careers researcher in some of the current projects to show us something about their research. But I wanted to just mention some of the earlier projects. For instance, Eiko Yoneki and John Crowcroft's flu phone was a very, very early use of phones to track people with flu. And obviously this has become a very interesting hot topic in recent events. Cecilia Mascolo's work, her team's work on COVID sounds is represented here. But before that, there's some groundbreaking work on mental health and using phones and so on to see how people with various mental health issues were dealing with things in terms of how they were moving around in the university location and so on. And Matea Jamnik and Pietro Leo, I could mention their work on cancer research. So this is a whole range of different types of topics. What we're hearing today, as I said, is an opportunity for the early career researchers to present their work. And we're very keen for people who hear this, get excited about this, and maybe want to work with us on perhaps on other topics. So Helen would be the right contact for that, or one of the speakers if that's more appropriate. So I want to finish this welcome by saying thank you very much for participating. We had over 150 people who registered from all areas, including not just academics, but also industry and, in fact, clinicians. And so we're really thrilled to have you all here. And yes, and thank you very much to Helen Francis and her team for organising that event. So I'll hand back to Helen now. Thank you.
Thank you, Anne. I'm now going to introduce our first speaker, Dr. Sarah Morgan, who's going to talk about predicting outcomes for psychotic disorders using brain connectivity and transcribed speech data. Over to you, Sarah.
Great, thank you, Helen. So my name is Sarah Morgan. I am a research fellow at the Computer Lab, and I am also working as part of the Accelerate Science programme which is all about sort of translating tools from machine learning into different domains of science. So I'm going to talk today about my research predicting outcomes for psychotic disorders using both brain connectivity and transcribed speech data. So psychotic disorders tend to develop in early adulthood, so normally when people are sort of in their early 20s or so, and approximately 25% of incident cases, so people who show sort of early psychotic symptoms such as hallucinations or delusions, go on to develop chronic conditions such as schizophrenia. But to date, there are no reliable quantitative methods to predict outcome for individual patients for psychosis. So we don't know which of the people who first develop those early psychotic symptoms will go on to develop really some conditions like schizophrenia, which are really some of the most debilitating mental health conditions. So the aim of my research is to better understand and predict psychotic disorders using both brain networks derived from magnetic resonance imaging. So these are sort of estimates of brain connectivity, which we can get from magnetic resonance images, and also transcribed speech data. So I'm going to start by talking about some of the work that I'm doing around brain connectivity. So brain connectivity is known to be disrupted in psychotic disorders. That's been clear from about the last sort of 10, 15 years of research, particularly using MRI imaging. But it hasn't always been clear sort of which brain connectivity features are really disrupted or altered in schizophrenia. So to try to find brain connectivity features, which ultimately we hope may be predictive of individual disease trajectories, we started with the sort of simpler question of looking for sort of which brain connectivity features can replicably distinguish patients with sort of chronic full-blown schizophrenia from healthy volunteers. So starting with that sort of easier question. So we trained machine learning classifiers to do this using Gaussian processors on 3 independent data sets. I found that we can do this classification problem with sort of reasonable accuracies, 70 or 80% accuracy, a little bit data set dependent. But most importantly for us, those machine learning classifiers were really sort of driven by a very replicable pattern of disrupted brain connectivity, which was sort of replicable across those 3 independent data sets. So you can see the brain connectivity features that are diagnostic of schizophrenia are really quite replicable. So we think now we've built up this understanding of sort of which brain connectivity features are really diagnostic for schizophrenia. We've also done some work then trying to relate this sort of pattern of altered brain connectivity to underlying gene expression data to try to build a better picture really of sort of what the biological mechanisms underlying this disrupted connectivity might be. So I won't say too much about that, but we would be very happy to answer questions about this or talk more about it if people are interested. Really what we're doing here is using gene expression data as a sort of map to understand what's going on in the brain at different brain regions, and to understand some of the biological mechanisms that might be underpinning the disrupted brain connectivity we see. The next steps for us are then trying to understand, given that we now have I think, quite a good picture of where brain connectivity is disrupted in schizophrenia, can we then use those brain connectivity signals to try to predict longitudinal disease outcomes for individual patients? And to do that, I'm working as part of a large consortium called SciScan, which is a sort of multi-site EU-funded project. And at the moment, we're collecting data at sites around the world to try to get that data to be able to predict individual outcomes. The other area of my work is really looking at speech markers of psychotic disorders. So incoherent or disorganized speech has long been known to be a core symptom of psychotic disorders. And recently we've shown that that could be captured by quite a wide range of different natural language processing markers. So from very simple things like just number of words, which is reduced in patients with first episode psychosis, shown here in yellow, compared to healthy control subjects, shown in blue, through to more sort of involved natural language processing markers, such as semantic coherence, which is again reduced in patients compared to healthy controls. And you can see, this is just sort of zooming in on this coherence, semantic coherence measure. You can see that that measure is really actually remarkably discriminative between healthy controls and patients with psychosis. And here in the center, you've got a group of people at clinical high risk of developing psychosis, which have these sort of intermediate values. We're now working on a new sort of speech graph tool to try to map out the semantic content of speech from psychosis patients in more detail. So, for example, this is a speech excerpt from a patient with psychosis and describing this image here. And so we're really sort of trying to map out the speech, the semantic content of this speech using a network approach. And this work's being led by Dr. Caroline Netterkoven, who's a postdoc working with me on this. So to summarize, brain connectivity and speech show replicable differences between patients with psychosis and healthy volunteers. And we've done a lot of work over the last few years, really trying to map and quantify those differences in detail. The next steps then for us are to try and use these markers to predict outcome for individual patients in longitudinal data sets. So thank you very much for listening. I'd be very happy to take any questions.
Thank you very much, Sarah. So as Sarah mentioned, please feel free to ask any questions by typing them in the chat, and then I can read them out. I'd like to start off with a question, Sarah. How do you get from the systems you described to tools for clinicians to use, and how long will this take?
I think that's a really good question. I think the answer is probably somewhat different for the speech, transcribed speech work that I'm doing compared to the MRI work. So I think the transcribed speech work is potentially closer to clinical application. At the moment, the sort of barriers there to clinical applications are that it's quite a new field. Generally, clinicians don't routinely collect speech data from patients with psychotic disorders. So the sample sizes that we have at the moment are quite small. So at the moment, we're starting to look at how we can collect more data, including collecting data routinely in Cambridge, in the clinics that we have in Cambridge. So I think that's the sort of the main boundary there, so the main barrier there. But potentially, the signals that we get from the speech data are really quite strong, and I think we're starting to see how those might be translated in the not so far distant future. For magnetic resonance imaging, obviously the signals that we get are somewhat more subtle, but of course MRI gives us this fantastic opportunity to really see inside patients' brains and to understand some of the altered biological mechanisms. So I think for MRI, probably the main application is in a better understanding of the biological mechanisms that are being disrupted. Although, of course, there is still work also trying to build better sort of tools for prediction of outcome, which is another potential application, but maybe a little bit more longer term.
Thank you. And now we've had a couple of questions come in. Can you please provide more of the details of the methodology and NLP techniques you used?
Sure. So we're using a few different measures. Our sort of approach has been to try to look at a wide range of NLP techniques and approaches, and basically to see what works best. So I showed you some results using semantic coherence, so that's using word embeddings and then calculating coherence from those. As I say, at the moment, we're particularly interested in developing more sort of network approaches. At the moment, people have looked a little bit at sort of what they call more sort of syntactic speech networks, graphs really, where people take the sort of words in the speech and then build what's called a co-occurrence network, where the words become nodes and you link the words in the order that they're spoken. And those graphs show some alterations in patients with psychosis compared to healthy volunteers. We're trying now to build these more sort of semantic speech graphs, really to map the semantic content of the speech. And that's our main area of travel at the moment. Okay.
And then one final question. What software systems do you use for the machine learning aspect?
So at the moment, for the MRI work, we've been using Gaussian processes for classification of cases and controls. Those work quite well. They're also probabilistic, which is quite useful for us because it means that we can look to see whether or not the probability that a person is classified as a patient correlates with something else, either particularly of interest, so for example, whether or not that correlates with symptom scores, which might be what you expect, but also to check that there aren't sort of extra compounds, for example, with MRI, for example, that there aren't correlations with how much the person moved in the scanner, which is something that we're not so interested in.
Okay, thank you. And we just have one more question, actually. Are there obvious connectivity related conditions to explore after schizophrenia?
So yes, I think definitely there are many other mental health conditions that we could look at. Obviously in Cambridge people are already working on other conditions. Quite a lot to the speech data, there's quite a lot of work already on Alzheimer's disease. And that's quite an interesting avenue. So brain connectivity, people look particularly at developmental disorders in children, for example. I think that's a sort of one particularly obvious avenue to try next.
Okay. Right, thank you. Oh, and one, we've just got time for the last one. Do you see any potential for the speech technologies to be incorporated into the likes of Siri and Alexa to diagnose patients in the future?
Potentially. I think in the first place, we will try to get these tools working in clinic, particularly where patients are answering, particularly where patients are given a task where they're describing, for example, a picture, and we can record speech in a much more sort of controlled way. That also avoids a lot of the issues around sort of ethics, potentially recording information that you don't want to record, or personal identifiable information, etc. So I think longer term, there are many ways that you can imagine, particularly the speech work being applied, but obviously there are lots of ethical challenges too.
Okay, thank you. I think that's all the time we've got for questions now. So thank you very much, Sarah. I would now like to introduce our next speaker, Dr. Indu Bodala, who's going to talk about her work on designing a robotic mindfulness coach, a longitudinal study.
Hello, everyone. Thank you for joining me today in this talk about designing robot well-being coaches. I would like to start by providing an introduction about social assistive robots, or SARs, that are gaining prominence in the healthcare industry these days. For example, on the left, you can see a Paro robot being used as a companion robot for elders in care homes. And on the right is a QT robot delivering therapeutic interventions to children with ASD. However, the use and acceptance of these robots pose several challenges, such as design aspects like appearance, embodiment, and personality exhibited by the robot can influence the way it is being accepted, varying user expectations, and therapists or caregivers lacking confidence in these robots to use them for delivering the interventions, and of course, security and ethical concerns raised by various stakeholders. Using Wizard of Oz style investigation in human-robot interaction studies, roboticists can identify these problems and challenges right from the early stages of robot development. Robot teleoperation is one such method that can enable multimodal embodied interactions during human-robot interaction experiments. So motivated by this, we also use teleoperated Pepper robot for our research. Social robots are being used recently to deliver mental well-being interventions. For example, Kid et al. Used Autumn robot to instill behavioral changes in people while dieting. And Jiang et al. Recently used Jibo robotic platform to deliver positive psychology intervention to college students. However, current studies lack an investigation into how to deliver these interventions successfully using robot coaches in place of expert human coaches. This requires comparing the differences in interactions participants have with humans versus robot coaches. It is also important to conduct longitudinal investigation to ensure that there is no novelty effect during these interactions with the robot, and the robot is actually relevant for long-term use. So we plan to address these questions through our research. Our vision is actually to create an autonomous well-being coach that can exhibit embodied multimodal interactions for long-term and personalized use. Using robots for well-being interventions is a multidisciplinary research problem, and we adopt iterative design approach where we learn from the expert human coaches on how to deliver these interventions correctly, as well as collect interaction data and design requirements through face-to-face studies. For this purpose, we conducted a 5-week study where 2 groups of participants received mindfulness training course from an expert human coach as well as a teleoperated robot coach. During the teleoperation sessions with the robot coach, the human coach is located remotely and he teleoperated the robot using 3 important pipelines. The pose replication pipeline was used to relay estimated pose from the human coach to the robot in real time. We also implemented vision and audio pipelines so that the human coach can see and talk to the participants remotely through the robot. So after each session, the participants filled a session experience questionnaire where they rated the human and the robot coaches on the following items. We also collected participant personality scores using 20-item personality survey. All the sessions were recorded to gather facial and audio data. And we conducted a longitudinal statistical analysis on the interaction data to understand the interactions people had with human versus teleoperated robot coach. During the interactions with the human coach, the participants rated the coach highly for all the weeks in all the aspects. So this suggests that they reacted positively and confidently towards an experienced human coach. However, during the interactions with the teleoperated robot coach, low ratings were given for the aspects of animacy and anthropomorphism, while the ratings are still high for the likability due to the appearance of Pepper and perceived intelligence as it is being teleoperated by a human. We also found significant increase in the ratings of robot motion as well as conversation aspects with time, suggesting that participants may need some time to understand the robot capabilities and improve their perceptions towards these aspects. We also compared the ratings on the scale of anxious, relaxed, agitated, calm before and after each sessions. We found that each session promoted significantly positive mood in the participants for both human and robot coaching. Moreover, less variability was observed for robot coaching sessions compared to human coaching session ratings. This may be due to the fact that human-human interactions are more prone to variations in nonverbal and expressivity behaviors that can lead to more variability in the participant ratings. However, this is not the case with the robot. So this fact actually needs further investigation. Along the 5 personality traits that we observed from the participants, we found that conscientiousness and neuroticism scores of participants had significant associations with the perception ratings given to the robot coach. A negative association between conscientiousness and robot motion was found, suggesting that high conscientious people expected the robot to move more naturally than it currently does. People gaining high neuroticism scores also rated the robot low when they were asked about how much they enjoyed the sessions. So these results suggest that the design of a robot well-being coach requires personalization along the personality aspects as well. So our work thus involves gaining insights into how participants perceive the robot mindfulness coach with time and how their subject-specific characteristics also influence these ratings. In our ongoing work, we aim to take more qualitative approach in gathering the design requirements through unstructured yet effective formats of interviews using participatory design studies. We also aim to develop effective human-robot interaction models guided by human-human interactions, which can be further applied for generating autonomous robot behaviors. In the future, we also aim to explore more on multimodal synchrony as well as supervised autonomy for designing our autonomous robot coaches. I would like to end today's talk by extending my thanks to my co-authors as well as lab members who have helped me conduct this research. Thank you very much for your attention, and I'll be happy to receive any feedback or questions from you now.
Okay, thank you very much, Indu. That was really interesting. So, as I said, we'll take some questions and answers through Zoom. I've got a question to kick things off. So I imagine that doing the kind of assessments you discussed remotely has been even more relevant with COVID. Have these circumstances influenced the research at all?
Yes. So thank you for the question. So telepresence robots are already providing some effective solutions due to the pandemic situations. For example, we have seen telepresence robots giving virtual museum tours or being used to create in-proxy presence of closed ones for older adults in care homes. We are also in our research, we are also exploring the use of telepresence robots for safer HRI studies, where participants can just interact with the robots alone without coming into close contact with either the experimenter or the human coach during these experiments. So there is quite a lot of research going on using telepresence robots to tackle the pandemic situation.
Okay. Okay, thank you. We've had a question come in on the Q&A. What are the latency requirements for teleoperation?
So we are actually able to replicate almost near real time. For example, just for the pose replication, we have around 7 frames per second replication, and then that will be interpolated in some smart way to make it more smoother and continuous. We are also applying some constraints on the angles, joint angles projected on the robot real time to avoid any dangerous or harmful movements. So yeah, it is quite smooth for the implementation. Yes.
Okay, thank you. You mentioned personalizing the robot to reflect different personality traits, but how easy is it to do that?
As shown in our study as well as in the previous research, we found that the personality exhibited by the robot can actually influence participants' perceptions and also their understanding of the robot design and behavior. So if, for example, the previous studies in our lab group, we have attempted for automatic personality prediction of the participants, and then also manipulate robots' behavior on the scales of extroversion, introversion, and we found that there is a difference in the engagement levels of the participants with the robot based upon what kind of behaviors are exhibited by the robot. So so it is possible in some aspects of the personality. However, further research is needed in other aspects of personality, for example, openness or neuroticism. So we still have to understand how to manipulate robot behaviors in those scales.
Okay, thank you very much, Indu. I think that's all the questions that we've had in. So thank you.
Thank you so much.
Okay, we're now going to hand over to Dr. Jing Han, who's going to talk about her work on automatic COVID-19 diagnosis from breathing, cough sounds, and voice.
Thank you, Helen. Hello, everyone. I'm Jing Han. I'm a postdoctoral researcher from Mobile System Group, and today I'm going to talk about one of our projects about audio-based COVID-19 diagnosis from breathing, cough sound, and voice. First, I would like to take a moment to introduce our team members for this project. They are researchers from different expertise for mobile systems, respiratory biology, audio analysis, and machine learning, of course. And all these researchers have contributed to the work I'm going to introduce in the following. As we know that early diagnosis for coronavirus is quite critical for timely treatment of the patient, as well as for effective control of the disease spread. And currently, the standard way is the swab test. However, in the real application, has some barriers prevented for a large-scale detection. For instance, that it requires high workload and needs skillful operators for the sample collection and testing. And also, the instruments can be quite expensive and need special operation places. So the aim of our research is that if we could develop a test which can be available almost to anyone, anywhere, and anytime. And our vision is to build a audio-based smartphone application which can collect the audio samples from individuals via smartphone and then give the feedback about their test results. The background of this research is that, as we know, there are a wide range of symptoms reported for the disease, and many of these symptoms are high correlated with human sounds, or say can be perceived from sounds. For instance, these are listed here in bold, including continuous cough, hoarseness with shortness of breath, and the last one is the dysphonia, which indicates difficulty in speaking due to some disorders. So for this aim, our group developed software, developed smartphone application where the participants can download the app and follow some instructions. They can provide 3 coughs, 5 breathings, 3 voice samples, together with other basic information such as their age, gender, and their symptoms, the latest COVID test results, if any, together with medical history, smoking history, and we also collect the location information if the user give consent to us. The pipeline of our work is, once we collect this crowdsourced data, which could be very noisy, we need a stage for data preprocessing, where we use the different technologies for data cleansing, label cleaning. By different data selection criteria, we will also need to control the quality of the audio sample, and we also do data segmentation to remove the silence part from the beginning and the end of each recording. Then the last 2 stages are quite standard machine learning stages, where we investigated handcrafted features, acoustic features, as well as some data-driven high-level representations. And then for the classification task, where we detect the COVID or not, and this can power other following applications such as screening or monitoring in the tragedy of the disease or for risk assessment. Since we launched the app in April last year, in the first 8 months, we have collected more than 30,000 samples from 15K plus users. Most of them are male, and most users are between the age from 20 to 50 years old, and most of them are non-smoker. And these are the distribution of the data we collected with respect to their COVID test result. And then in the last 3 months, from November till the end of January, the data we collected are doubled, meaning that now we have get more than 60,000 samples. However, do note that when comparing the COVID test results labels, most of these users do not have the COVID test results. So only a small portion of our data are with either positive or negative COVID test results as our labels. So here we show the current model we built for this audio-based COVID diagnosis. So we use convolutional neural network to learn high-level representations from cough, breathing, and voice sounds separately, but jointly concatenate these higher representations and feed them into another 2 dense layer to derive the final scores. This score can be used or interpreted into 0 and 1 for a binary diagnostic outcome for COVID detection, or this score in the range of 0 to 1 can be deemed as the probability for a longitudinal diagnostic progression. Currently, for binary classification tasks on a holdout test set, our model achieved the performance in terms of error under the ROC curve about 0.77, and with a sensitivity of 0.74 and specificity of around 0.68. We do note that our performance are stable across different age and gender groups, and there is no obvious variance across different health conditions, such as for asthma patients or for different smokers. Then for the longitudinal study, here we show some examples. For example, the first one where the axis indicates the date where the participant provide a recording, and the y-axis indicated how our model estimated the possibility that the audio sample or the user are of COVID positive. So here for the first example, our model picked correctly at the beginning. The patient was with a positive label, and then along the time we could see that the probability decreased, indicating that this user are towards recovery, healthy again. And then for the second participant we chose here, who provided the 5 negative COVID test samples, our model's prediction are close to 0, indicating it predicted negative for this participant. Then for the third user we chose here, this NT stands for not test yet. So our model kinds of predict earlier, like 2 days earlier than the real positive test claimed by this user. However, at that time, our model's probability for positive prediction is decreased, and we lack information whether this indicates our model failed at this time, or that actually the user get the positive test results might take a while from the real onset of the disease. Or the last example we showed here is somehow our model predicted correctly, like 1 day later than the self-reported positive test result, and also show some fluctuation at the beginning. Also shows that our model needs further investigation to improve in this longitudinal study. Last but not least, Here I point out some future research directions we are working on. So the first aspect is for data exploration. Recall that currently we only focus on the data with valid positive or negative test results. However, the other data without this test result can still be valuable, and we need to deal with the noisy label as well. And currently, to guarantee a good performance, we do manually check the quality of the audio sample. But once it goes to large scale, we need to automate this chain. Then for the disease screening, apart from further improve the accuracy and the robustness of our model, another important aspect is to increase the interpretability of our model, so make the decision more transparent and reliable, and make it easy acceptable for the public or for clinicians. Then for the disease progression, we need to further investigate the potential for our model for the early detection of the disease, and there is the necessity to implement the user adaptation for this personalization to take the individual differences into account. And also it could be interesting to investigate whether there are some long-term effects of the disease on human sound. Then hopefully once we conquer this disease in the post-COVID era, we could still use this data for other research, such as in our data, we have users who has asthma and other respiratory disease, and we could use this data to study the correlation between the sound and other symptoms. That's it. So thank you for listening. And if you feel interested in our project, you are welcome to scan this QR code and download the app. Thank you.
Okay. Thank you very much, Jing. That was brilliant. So I've got a few questions for you here. Firstly, have you got enough data yet to be able to compare different languages and different ethnic backgrounds in the samples that you've got?
Yeah, so this is a quite good question. So as you know, the data are crowdsourced, so the data can be very noisy, and currently we do the quality control to get rid of very noisy data, like some recordings has the music or the TV played in the background. So that kind of data at this early stage, we just get rid of these noise samples. And then for the language aspect, as we noticed that we collected the data from different languages, and actually the people are requested to repeat one sentence 3 times. So the content somehow from within one language is fixed, but across different languages, this could still affect the acoustic features analysis from this aspect. We aim to develop a language invariant model, so which can make robust predictions across different language, but that needs less biased the data during training. And currently, we are still short of some other language data beyond English.
Okay, thank you. And a question here has come in. Could different microphones on different devices affect the reliability of eventually detecting if someone's got COVID? So you mentioned you obviously dismissed some noisy samples and that kind of thing, but could this have an effect on whether you determine someone has COVID or not, and how do you account for it?
So currently, yeah, we have different version of the app, and because the data are collected from different devices, so there will be different sampling rate. And what we currently do is that we resample all these things and try to get try to mitigate the effect of different devices. However, I should say that this is quite challenging task, and we still need further investigation. And another thing we plan to do is like to embed the function of the uncertainty of our model, so that if the data distribution between the training data and the inference are too large, then in such case, when our model are less confident to provide a prediction for the different for this sample with such difference, then the model should refuse to give a decision for such kind of samples.
Okay. What kind of audio features I know you detect various different audio features, but which of the ones that you're using seem to be most relevant for detecting the positive cases?
We looked at both the handcrafted acoustic features. So actually we did some data analysis, try to find whether there are some distinguishable acoustic features which can identify COVID, and there are some other research groups tried looking into that direction, and they find that also in our data we have seen such acoustic features like the fundamental frequency standard deviation. Like when people get infected by the disease, it seems that they are less likely to control well of their voice, so that we see more disturbation in their voice. And also for the cough and breathing, the energy seems to diffuse across more frequency band. But then the model we introduced here, which used the spectrogram directly, actually it didn't really explore the acoustic features, the handcrafted acoustic features directly, but rather try to learn the data-driven features. And we used a pre-trained VGG convolutional neural network structure to leverage the power of big data from other audio samples, and then use this to fine-tune our model with our own audio data.
Okay. Another kind of related question here. So how can you account for the differences, and how easy is it to account for the differences with the samples you've got from a very bad cough, like something you get with pneumonia, compared to COVID?
So currently what we do is that we train a quite general classifier where we are not just distinguish from COVID cough versus a healthy cough without any other disease. But what we do is a general task where doesn't matter whether this person are symptomatic or asymptomatic, a COVID patient versus other non-COVID sound or samples, where we also have non-COVID but flu-like samples and with other symptoms. So currently we do not see that we have a bias, or say we behave our model predict bad when the models when we feed in asthma or other respiratory disease samples. But for these subgroups, we still manage to distinguish between COVID positive asthma patients versus COVID negative asthma patients.
Okay. Thank you. Right, I think it's time for us to move on to the next presentation. So thank you very much, Jing. That was really good.
Thank you.
So now we're going on to our final longer talk, and this is integrative and interpretable AI methods and tools for bench-to-bedside research in cancer medicine. And this is going to be given by Zohreh, Helena and Nikola. So I'm going to hand over, and I believe it's Helena who's going to speak first.
Hello, everyone. So thank you for attending the session. So in the next 20 minutes or so, Zohreh, Nikola, and myself will be giving an overview of the research we're doing here at the Computer Lab on cancer integrative medicine. So I'd like to start the talk by sharing with you our vision on where AI and medicine come together. So we've just heard actually some very interesting applications of computer science or AI into medicine, but we also believe that there's a lot on where medicine can contribute as well into the AI community. And more especially in the last years, we've seen how medicine has in a way been shaping so medicine has been shaping AI and helping develop some very novel techniques and methods. So hopefully we'll convince you of that in the next few slides. So we work on oncological medicine. So we're working on cancer, and we're part of this bigger project, which is called the Mark Foundation for Cancer Integrative Medicine. So this is a big consortium that brings together experts working on cancer from many different angles. We collaborate directly with doctors and clinicians that are treating patients on different types of cancer. So from breast cancer to ovarian, renal, etc. But we also collaborate with experts that have knowledge on different techniques to diagnose and to treat cancer, for instance, like radiogenomics, so some other different types of imaging techniques. And so the idea of the Mark Foundation is to bring together all this expertise and all this knowledge to build a platform for better diagnosis and to improve diagnostics and to build more personalized treatments for patients. So yes, so our part on this project, our goal is to bring together all these different types of knowledge, all these different heterogeneous patient data, and integrate it also with external knowledge to, as I mentioned, build more personalized diagnostics and treatment. And we're currently working with breast cancer data, but our methodologies are general enough that they can be applied to other different cancer types. So to be a bit more specific on which clinical targets and which applications can benefit from integration. So these are some examples. So we work on trying to predict treatment response. So how each patient responds to the treatment that they've been given. But then we also try to find out new ways of classifying maybe the tumor types, the cancers, or also to also improve the patient clustering that's already been done. We also work on predicting chem-induced toxicities, which is the response that the patient have when they've been given a drug. And we're also building patient and genetic profile graphs. So hopefully all of this sounds interesting, but when it comes to practice, there are several challenges that we are facing when we're dealing with healthcare and medical data. So some of this, or the main ones, would be, for instance, that we have very heterogeneous and high dimensional data. So as I said before, we're trying to integrate different types of data modalities. So we're working from genetic to medical records and also images. So all of these traditionally have been implemented or have been analyzed by using different techniques. So we're trying to combine them all to build a general prediction. And then on top of that, they all have very high dimensional, a very high number of variables. So for instance, images have lots of pixels. We have also a very large number of genes. So on top of this, it's not only heterogeneous and high dimensional data, but we also are dealing with low data regimes. And this is a problem that is common in many, many different medical applications. So what we mean by low data regimes is if we compare them with more traditional, for example, computer vision techniques that deal with images, those models are usually trained with data sets in the order of hundreds of thousands, if not millions of images. But in our case, our medical studies, we're lucky if we have in the order of hundreds or maybe thousands of patients. So this is actually a vital part of the way we develop our algorithms, so they can handle being in these low data regimes. And then finally, another big point that we try to make every time we're designing or developing a new model is to make our models interpretable and explainable. So we're not only trying to achieve high accuracies, but we also want to be able to understand the outputs and to understand how the models get to this output so we can communicate that to the clinicians and to the doctors that will be using them. So just before we go more into the methodology part, I just want to give a bit of an insight on the data that we're using and we're integrating. So we have three main modalities. We have genomic, clinical, and images. And the genomic side, we have gene expression, mRNA, and then copy number alteration, which are the mutations. These are usually tabular data coming in numerical form. Then we also have medical records for each patient. These are usually categorical or numerical variables, sometimes even time series. And then finally, we also have images, from where we also have different types of images. So we've succeeded in integrating digital pathology slides. So there are slices of the tumor where we can actually see individual cells and classify these cells into cancers, stroma cells, lymphocytes, then use that information to integrate with the other modalities. And now we're looking at integrating also MRI and mammograms. So MRI and mammograms have traditionally been analyzed by using radiomic techniques. So we are now looking at incorporating these radiomic techniques into the AI pipelines that we have for multimodal integration. So this is the data that comes from the patients, but now we also use external knowledge. So this is knowledge that has been curated from years and years of research, and it helps us tackle some of the challenges that I mentioned before. So, for example, we use protein-protein interaction networks, PPIs, and usually we've been using them in graph neural networks, but they mainly help us with feature selection and dealing with this high-dimensional problem. But then we also have gene ontologies, or we use gene ontologies for explainability and interpretability. And then finally, the last point on the data is the data sets that we're using. So we have two different types. The first one are the retrospective data sets, which would be Metabric and TCGA. So is that, okay, well, I'll just continue. So we have Metabric and TCGA. So these two are publicly available data sets, are some of the biggest ones that are already available right now. And we use them mainly for training and validation of our models. But we also work with smaller data sets, which are prospective. In some examples would be partner or the toxicities ones. And for these ones, we work directly with the clinicians that are collecting all this data, that are recruiting more and more patients. So these are ongoing studies. So we're deploying our models there, but also the data is increasing with time. So just to finish up, I'll give you an overview of our approach and our research line. So this is a 3-year project. We started in the first year by working on the integration side of our research. So we integrated genomic and clinical data to start with by using unsupervised learning and generative methods. And now we've succeeded also in integrating digital pathology slides, so images as well. And then on the second year, we started another research line, which is more on the interpretability and explainability side of things. And we developed REM, rule extraction models. So these are methods that allowed us to extract sets of rules from deep learning. And then finally, now we're going into the deployment of all of these methodologies. So we're developing a visual interface for clinical practice. So these are 3 research lines that they basically all work together, and they're all still ongoing. So we're extending them all in different ways. But I'll now just pass it to Nikola so he can explain a bit more on the integrative side of our research. Thank you.
Thanks, Helena. Next slide, okay. So as Helena mentioned, what we are trying to do here is to integrate different data coming from different modalities. However, as she mentioned before, this data also comes with different complexities, such as high dimensionality. So what we are trying to do here is develop methods that tackle these challenges together. So in a word, we're trying to learn integrative representations that on one side, we learn representations of the data itself in terms of the distributions where the data come from, and also these latent representations that we're trying to learn are also integrating this data. And in order to do this, we're sort of incorporating generative unsupervised methods such as variational autoencoders, which have some good properties that we can work with. Among some are that they give us the ability to learn this kind of integrations independent of the data analysis tasks that we have, and use this further down in different analyses that we're interested in. Next slide, please. And in order to do that, we of course needed to evaluate and experiment with different variations of how we can design this and develop this. So we performed extensive analysis, and we found that we can perform successful integration, in lack of a better word, where comparing to other benchmark methods, we were able to get much more accurate performance in terms of predicting different cancer phenotypes, such as distance relapse in breast cancer patients and different cancer subtypes. Now, of course, we barely scratched the surface of this. So what we are working now is along several dimensions. One is sort of integrating more data, which I will touch upon in the next few slides, where we are trying to integrate image data. We are also interested in improving the learning paradigm behind how we train these variational autoencoders, and we are investigating in different aspects of, I don't know, from different regularization mechanisms to how the learning procedure is performed. And of course, we're also interested in more experiments where we want to sort of establish the findings that we had before on different cohorts and on different predictive tasks. However, these variational encoders, it is important to say that while we can do integration, they're still far from the interpretability side of things where we want to be. Next slide, please. Now, in order to add more data in this whole story, we started with digital pathology images. So from biological point of view, digital pathology images are absolutely crucial in terms of identifying really important prognostic markers, and in turn help with different predictive tasks such as survival analysis of cancer patients. And we've seen throughout the years that from machine learning point of view, they have been used before and have provided really sort of good insights into the whole process of the disease. Now, the idea here is that we want to integrate this with the omic data and the clinical phenotype data that we have together in order to see whether we can even get even more performance out of it. Since we want to think of them as sort of like an intermediate bridge between standard radiological images that one has on one hand side and the omics data on the other hand side. Now, I mentioned that the variational autoencoders that we developed before are sort of a bit far from the interpretability side of things. So here we take a bit of a different approach. So instead of feeding just raw images into the autoencoders, we also go through a step of image segmentation and feature extraction out of the images first before combining them with clinical phenotype data and multi-omics data. Next slide, please. And we found out that integrating this data indeed leads to better performance, especially when it comes to non-trivial tasks such as distance relapse in cancer patients. And of course, this step that we took towards interpretability paid off, since now we can also investigate what kind of features that we've extracted in the first phase yield to better accuracy and give sort of some kind of a feedback to us as let's say machine learning modelers, as well as to the clinicians, so they will know what to look for in the future. And of course, we haven't stopped there. What we're working on now is trying to see whether we can include more image data, as well as trying to see whether we can improve this first step of image segmentation by resorting to other methods that we can use for better, let's say, image segmentation when combining different data, such as radiomics data. Next slide, please. However, what I've talked so far is only on the data side of things. Now, we want to make another step forward towards the interpretability by integrating knowledge. Next slide. So again, from machine learning point of view, it's been a long tradition of analyzing gene expression data in order for understanding the molecular status of a cancer. However, from machine learning point of view, this has always been leading to different kinds of problems in terms of overfitting models, and sometimes even sometimes even results that are not sort of captured in the data itself. Now, what we are trying to do here is to find ways of how we can bring existing domain knowledge into the picture, and we're doing this by actually trying to embed the structure from known protein-protein interaction networks into construction of computational graphs. Next slide, please. The idea here is that we're trying to use biology as a computational formalism when we are trying to develop a novel predictive models. And by predictive models here, we think of neural networks. So instead of using the topology of the PPI networks as a constraint in terms of feature selection, we use it towards designing or learning the computational graph of these predictive models. And we found out that this approach indeed leads to better performance when compared to other benchmarks such as, I don't know, fully connected neural networks or even randomly connected neural networks. It's readily generalizable to other tasks. So when we design these computational graphs, it doesn't need to be specific to a specific cancer predictive task, but it can be generalized to different ones. So this sort of proves as a promising avenue. And what we're working on now is figuring out whether we can sort of combine more knowledge and see whether that can take us. And of course, also experimenting with other cohorts and graphs. And for the final steps towards interpretability, I will just pass to Zohreh so she can tell you more about the interpretable side of things. Thank you.
Thanks very much, Nikola. So yeah, we heard from Helena and Nikola regarding different data that we've got available to us and also the cool integration methods that we have used. But of course, one thing that is crucial is having the kind of eventual deployment of this in mind. So in terms of deployment in clinical decision support system, or rather ensuring that deployment is possible, we have identified two main pillars, one explainability and one clinical accessibility. For explainability side of things, our approach has been to kind of provide an integration of machine learning and machine reasoning to ensure the interpretability of our results, and on clinical accessibility, which is very much interlinked with explainability. What we have done so far is to make sure that our results are visualizable, and we can provide interactive interfaces where the clinician can actually interact with what we have designed and used. So what I would like to specifically talk about on explainability front, it's rule extraction methodologies, REM, that we have designed here in Cambridge. So if you look at the image on the right-hand side, we talked about different data modalities that might be subject to some processing. And most of the time we have ended up using deep neural networks for modeling and making predictions. So what we've done, it's basically to approximate the neural network with a rule set that can mimic the decisions of the neural network. And it's a decompositional approach, meaning that it uses both hidden features as well as the input features. And essentially the benefit of approximating the neural network, which is an uninterpretable model, with an interpretable model such as a rule set, is that then explanation is just readily available. And why we have chosen to approximate with rule set, two main reasons: because they're very close to what clinicians are used to, and also rule set are very easily integrable with other rules coming from other sources. This is not necessarily the case for some other interpretable models such as decision trees. So again, if you look at the right-hand side, we haven't made the assumption that we always model the data using a deep neural network. We have also assumed that the data can come from other sources that are best modeled with trees. And what we do is basically allow the combination of rules that come from neural network and trees to come together, plus the rules that come from the knowledge. And that way we provide a rule set model that is available for multimodal reasoning, basically, multimodality reasoning. And in terms of the REMD component of this, the red part of the figure, which is rule extraction methodology from deep neural networks, we have evaluated this extensively. We've shown that it can very efficiently approximate the neural network in a time and memory efficient manner. And we've also shown that the rule extracted the rule set model extracted is very compact. And it's just a very compact set of rules, which are tend to be very short in length. So I made a point about the integration part. One thing is that, so we've done all these things with the hope that it can help deployment in clinical decision support system. So now we want to talk about the part where we have come up with a set of functionalities for a REM that allows clinicians to easily interact with REM. So the main thing is that basically using these rule set from data knowledge, they can make a prediction. And not only that, they can also ask for an explanation of that prediction. What this basically facilitates, it's investigating the biological relevance of the rules and the prediction that they make. So say in the case of genetic data, they can easily look up at the features that come up in the rules and link them with existing bioinformatic tools, such as a gene enrichment analysis and pathway analysis, and make sure actually the model extracted is biologically relevant. Not only that, clinician can also adjust the rule base based on their tacit knowledge in their that they might have in their day-to-day clinical practice. So these are all the benefits that make REM accessible to clinician. But on accessibility front, it's not only REM. We have also done work on kind of more general ways of facilitating the uptake of machine learning methods by clinician. One particular project that I'd like to mention is called VIDA. Visual interface for integrative data analysis. So VIDA, it's more general in a sense, and it does basically allow clinicians to work with the input data, model it using some machine learning model, and also interact with the output of it. It allows exploration of the input. Basically, they don't need to do any encoding. They can just enter the parameter via GUI and fit a model to the data, and eventually the result is visualized in a way that is accessible to them, and they can potentially interact with the visualization as well. So on the covering kind of both explainability side and clinical accessibility, I think on this front, the whole aim is to ensure that deployment. And this brings me to kind of a summary of the talk, what we have talked about, all three of us so far, which can be summarized in 2 points. So basically, the aim was to provide this bench-to-bedside pipeline for personalized cancer medicine. And on the bench side, what we have done, we have developed this efficient integration method that allows integration of both data and knowledge. And on the bedside, we have made sure that this result can actually be brought to day-to-day clinical practice using knowledge-based AI approaches and also logic-based approaches, and kind of ensuring that the findings are interpretable. One thing that was touched upon, and apart from the extension that we are making in each stream, one thing that was touched upon was this low data regime, which is our current main open problem. And I'd like to kind of mention that as well. So Helena did mention the combination of low data regime with very high dimensional space, which is very common in oncology. We might get a data set that comes from a trial, and it might be 100 patients with thousands and thousands of features. I mean, for many machine learning algorithms that are data hungry, this is a very challenging task to work with such a data set. So to tackle this problem, we have tried several different approaches, some more conventional, some newer one, such as dimensionality reduction, using attention mechanism in graph neural network for convergence. But one thing that I'd like to mention that has proven particularly promising so far is to use biological ontologies, especially gene ontology and phenotype ontology, for feature selection. I won't have the time to go into details, but I just like to mention that basically what we have found so far is that if we have very little knowledge of few features that are related to a phenotype, we can use this ontology space to pick features that are relevant to the ones that we know and basically do dimensionality reduction or feature selection using that method and essentially have a good prediction even in a very low data regime when you have only 100 patients available to you. So I'd like to finish on the progress to date and the open problem that I just captured briefly. And on that note, I pass on to Nikola to finish off.
Yes, thanks, Zohreh. So in the final few moments, I just want to acknowledge that all of the work we did so far is tremendous collaboration between many, many brilliant people. So we would like to just acknowledge this, and we would like to say that for anyone who is out there and you want to also sort of contact us or just have some questions or you want to work with us, feel free to get in touch. And with that, I would like to thank everyone and ask if someone has questions. Thank you.
Thank you. That's really fascinating talk. I'll start off with a question actually for you, Nikola. So what you focus on so far is work on the kind of computational side, so developing the methods and approaches. How do you take this forward to be deployed in clinical practice? I think that's something a lot of people will be wondering.
Right. Well, yeah, that's sort of a good question and a really old one, right, for us, which are on the computational side of things. So as I mentioned on the very last slide, we're collaborating among those people that we're collaborating. We are also closely collaborating. Zohreh, can you go back a bit, please? With like the people. Anyway, so Zohreh. Okay, no worries. We are collaborating also with many oncologists. So everything sort of that we're developing, it's always with their guidance, and we're trying to facilitate everything together. So on that note, I would just say that that's sort of the first step. So collaboration with the oncologist, actually understanding their needs in a sense. The next thing is this whole pipeline of bench-side to bedside, where we try to sort of develop, in a sense, a holistic framework of how we treat data when it comes from a patient to sort of how it goes through the whole, let's say, machinery, and at the other end is taken by a clinician, and again presented. The results are sort of used for any personal treatment. It's, I think, how we can at least start thinking of getting this into sort of more day-to-day personal medicine practice. Other than that, I'm not a clinician nor decision maker, so this is sort of like my utopian view of it. But I'm really happy to discuss with anyone that can sort of answer it more, which is from the other side of the story.
Well, maybe there's someone listening who can help with it. Yeah.
I'm happy to discuss. Yeah.
Okay. Right. Moving on. A question. So Zohreh, we know that genetic data is really expensive, and it's not that easy to collect. Can computation help at all to provide personalized cancer medicine in the absence of access to this data?
Yeah, that's a very interesting question. We know that genetic is is at the core of personalized cancer medicine, yet it's very expensive to get, and it's not something that is a kind of mainstream in every hospital, say, in the country. So we have done some work on that front. We know that different modalities, such as, say, imaging, is very cheap to get, especially pathology images. We know that there are some connections about different modalities, say, between imaging and genetic. There is little known about it, but what we are trying to do is to kind of computationally investigate if we can predict some of the genetic features with imaging data that is very cheap to get. And if we can do that, then probably instead of having a whole genome sequence of the patient, prediction, we can try to predict those that are very essential for, say, a certain targeted treatment or certain part of survival analysis, and hopefully go around having access to that genetic data. And so far, we have done some prediction based on kind of a connection between imaging and genetic, as I said, but this could be trying to predict some genetic with clinical or other variable as well.
Okay, thank you. I just had a question come in. So how do you see this system implemented in the NHS? Is it a local facilities to process the data and a central engine to do the inference? And she's saying, I guess, that you're targeting having this implemented in as many sites as possible.
I'm happy to reply if anything.
Okay, thanks, Helena.
Yes. So in our case, like our models, as we said, they usually need lots of data to be trained, although we are dealing in low data regime already. But if we deploy them at a local level, there's the risk that we can't really train the models with enough data to be accurate enough. So probably we would be talking, this would be in the future, but we would be talking into having more of a centralized model and then potentially deploying it in, like, taking care also of all the privacy issues that may come up with it, but then deploying it at a local level.
Okay, thank you. Well, I think we are now out of time for questions. So I'd like to thank you very much for your presentation, and we'll just move on to a closing slide. Okay, yeah, so thank you very much to all the speakers. I think we've had some really fascinating presentations this afternoon. And thank you to everyone who has joined the event. It's the first of these events that we're going to run, and we're hoping this will become a more regular occurrence. If you do have any questions, please do feel free to get in touch with the speakers directly, or alternatively, you can contact the research strategy team directly. My email address is shown on the side, research strategy manager, and there's more information on the website as well. And if any of you were not able to join at the beginning or would like to go over any of the presentations again or share with colleagues, the recording will be available on our YouTube site. All that remains for me to say is I hope you enjoyed the event, and we will be sharing details of our next research showcase in the very near future. Okay, thank you very much.